Hi everyone, welcome to NEET Biology channel. Which one of the following is not a criterion for classification of fungi? A. Mode of nutrition. B. Mode of spore formation. C. Fruiting body. D. Morphology of mycelium. Correct answer is A. Mode of nutrition. All fungi are heterotrophic, and thus mode of nutrition does not provide diversity for classification. Fungal classification is primarily based on spore formation, mycelium morphology, type of fruiting body. Hence, nutrition is not a differentiating criterion. The equation of Verhulst-Pearl logistic growth is as follows. From this equation, K indicates A. Biotic potential. B. Carrying capacity. C. Population density. D. Intrinsic rate of natural increase. Correct answer is B. Carrying capacity. Explanation. In the logistic growth model, K represents the carrying capacity, the maximum population size that the environment can sustain. It accounts for resource limitations and causes growth to level off as population nears K. Which one of the following can be explained on the basis of Mendel's law of dominance? A. Out of one pair of factors, one is dominant and the other is recessive. B. Alleles do not show any expression, and both the characters appear as such in F generation. C. Factors occur in pairs in normal diploid plants. D. The discrete unit controlling a particular character is called factor. E. The expression of only one of the parental characters is found in a monohybrid cross. A, B, and C only. B, C, and D only. C, D, and E only. D, B, C, D, and E. Correct answer is C, A, C, D, and E only. Explanation. Mendel's law of dominance explains how one allele can mask the expression of another in heterozygous condition. Statements A. Dominance, C. Pairing, D. Factors, and E. Phenotypic dominance are true. B is incorrect as both traits do not appear in F2, only the dominant does in F1. Match list I with list 2. List I, tissue, list 2. A. Companion cell I dead, thick-walled, non-living. B. Guard cell 2. Living, thick-walled. C. Phloem fiber 3. Living, nucleus present. D. Trachid 4. Dead, elongated, pointed ends. A. A2, B3, C4, DI. B. A3, BI, C2, D4. C. A4, B2, C3, DI. D. A3, B2, CI, D4. Correct answer is D. A3, B2, CI, D4. Explanation A. Companion cells are living and have a nucleus. 3. B. Guard cells are living and thick-walled, controlling stomata. 2. C. Phloem fibers are dead, thick-walled. I. D. Trachids are dead, elongated with pointed ends. 4. Inhibition of succinic dehydrogenase enzyme by malonate is a classic example of A. feedback inhibition. B. Competitive inhibition. C. Enzyme activation. D. Cofactor inhibition. Correct answer is B. Competitive inhibition. Explanation. Malonate is structurally similar to succinate, the natural substrate for succinic dehydrogenase. It binds to the active site and competes with succinate, thus inhibiting the enzyme competitively. It does not alter the enzyme's active structure, making this a classic example of competitive inhibition. Formation of interfascicular cambium from fully developed parenchyma cells is an example of A. Redifferentiation. B. Dedifferentiation. C. Maturation. D. Differentiation. Correct answer is B. Dedifferentiation. Explanation. Dedifferentiation is the process by which mature cells revert to meristematic state to regain the ability to divide. Interfascicular cambium arises from parenchyma cells between vascular bundles. They regain the capacity to divide and form secondary tissues. A pink-flowered snapdragon plant was crossed with a red-flowered snapdragon plant. What type of phenotypes are expected in the progeny? A. Red-flowered as well as pink-flowered plants. B. Only pink-flowered plants. C. Red, pink as well as white-flowered plants. D. Only red-flowered plants. Correct answer is A. Red-flowered as well as pink-flowered plants. Explanation. This is an example of incomplete dominance, where the red, RR, and white, RR, alleles produce pink, RR, in heterozygous condition. When pink, RR, is crossed with red, RR, the progeny genotypes are 50% RR, red, and 50% RR, pink. So, both red and pink phenotypes appear. In a plant, black seed color, BB, is dominant over white seed color, BB. In order to find out the genotype of the black seed plant, with which of the following genotype will you cross it? A. BB. B. BB. C. BB. D. BB. Correct answer is A. BB. Explanation. To determine whether a black-seeded plant is BB or BB, you perform a test cross using a homozygous recessive, BB, plant. If all offspring are black, the plant is BB. If there's a mix of black and white, it's BB. Match list 1 with list 2. List I1, tissue structure. List 2, characteristic. A. Collenchyma 1 help in radial conduction of food. B. Companion cells 2, living with unevenly thickened walls. C. Sieve tubes 3, associated with sieve tubes and help in maintaining pressure gradient. D. Phloem parenchyma 4, enucleate cells. A2, B3, C4, D1. BA2, B3, C4, D1. CA3, B1, C2, D4. DA4, B2, C1, D3. Correct answer is BA2, B3, C4, D1. Explanation. Let's match correctly. A. Collenchyma, living, unevenly thickened walls, 2. B. Companion cells, help sieve tubes function, nucleated, 3. C. Sieve tubes enucleate, conduct food. 4. D. Phloem parenchyma, radial conduction of food. 1. The capacity to generate a whole plant from any cell of the plant is called A. Micropropagation, B. Differentiation, C. Somatic hybridization, D. Totipotency. Correct answer is D. Totipotency. Explanation. Totipotency refers to the ability of a single cell, like a parenchyma or meristem cell, to regenerate into an entire organism under suitable conditions. This principle underlies plant tissue culture techniques. Micropropagation uses totipotent cells, but is not the term for this capacity. How many molecules of ATP and NADPH are required for every molecule of carbon dioxide fixed in the Calvin cycle? A. 2 ATP and 2 NADPH. B. 3 ATP and 3 NADPH. C. 3 ATP and 2 NADPH. D. 2 ATP and 3 NADPH. Correct answer is C. 3 ATP and 2 NADPH. Explanation. For each carbon dioxide fixed, the Calvin cycle uses 3 molecules of ATP and 2 molecules of NADPH. These are used in the regeneration of RuBP and in the reduction of PGA to PGAL, G3P, respectively.